thrilled to be here. Thank you so much for inviting me. I think this is an awesome meeting. My crew is also always very worried when I go to SAGE meetings because I always come back with at least 3 collaborations. And I promised my crew this time that I will not initiate another collaboration until December, but January 1 is another year, right? So I was asked to be a little provocative, and so I chose to talk about orphan diseases since that is what I have been in since the last couple of years at the foundation, and I'll come back to that in a minute. But let me first say that I think in orphan disease there is a lot of optimism, but I also would like to add a little but to that. So first of all, the optimism is my first slide, is the yes slide. I think today rare disease is extremely popular. Just to define rare disease very quickly, for those that are not familiar, is below 200,000 Americans. There's about 7,000 different rare diseases. There is 30 million people in the United States that suffer from a rare disease, which is 10% of the population, and in Europe there is about 30 million cases. So we're looking at a very large population, but very diverse. Why do I think that rare disease is popular? Is because the FDA is really encouraging companies to enter into the rare disease space. And as we all know from the Orphan Drug Act, that has really led to incentivizing companies to enter into the rare disease space, as you can see from the couple of things that I'm listing now, but for time reasons, I'm not going to read them all, but let's say companies are really encouraged to enter the rare disease area. And if you look at the graphs, you really see a massive increase in drug approval since the Orphan Drug Act. So that is a first optimistic statement towards rare disease. The second thing is that the Center for Drug Evaluation and Research has really come up with a lot of innovative ways to stimulate drug development, and that has been extremely beneficial, specifically for the in the orphan rare disease space. You have the fast track, the breakthrough, priority review, accelerated review, all in fact aimed at the FDA that is now taking a very innovative step, I think, into the positive direction, helping companies to be fast, because of very often these rare disease people are in a space that are very often understudied, and it's very difficult to get endpoints. So the FDA is taking a much more active role here. And just to see what the effect is of that, if you look at the number of drugs in 2014 that were approved through those more innovative mechanisms, you can immediately see that if you look at the fast track, the breakthrough, priority review, and even if accelerated approval, all of the drugs that were approved in 2014 for rare disease were somehow in that, 8 out of 8 in accelerated approval were rare disease drugs, which I think is a very optimistic observation. The other thing that we see is that if you look at the money space, venture capitalists are increasingly interested in rare disease. I get very regular phone calls from companies and VC even that are interested in entering the rare disease space. So that is, let's say, the optimistic side of the situation. But there is, as usual, there is the but, right? We know, and Stephen and I have become very good friends because as Stephen loves to push people out of their comfort zone, I do that too. We know that we have a lot of typical roadblocks in the current R&D space, which are, I think, even more visible in the rare disease sector. And if I say roadblocks, I'm talking, in fact, about 2 big blocks of roadblocks. Roadblocks for the patients, especially in the rare disease space, is that there is often a problem of misdiagnosis. There was a report from Shire where they said that typically patients are almost misdiagnosed for 7 years before they know what they actually have. Patients are often undereducated. They don't know what their disease looks like. They don't know where the other patients are, and they feel very lonely. Lack of knowledge around patient needs. There is a limited access to clinical trial information and also clinical trial sites. And for the researchers and industry, we have the typical suspects, right? The tissue, especially in the rare disease space, is extremely scarce. The patient population is ill-defined, so that means that there is a lack of well-validated endpoints. The market is completely unknown. There is a lack of tools, which is a typical problem in R&D, but is even more prominent in the rare disease space. It's possible to buy the breast cancer cell line, but to buy neurofibromatosis cell lines is close to impossible. There is also a lack of data very often because there is a lack of researchers that are really in the field. And then we know that collaboration is sometimes an issue, but if you have a very limited number of researchers, that issue becomes even more important. So is that fixable? I am an optimist by nature, and I think it is, but there are certain things that we need to change. And we have a couple of success stories at the Children's Tumor Foundation that I thought were worth sharing with you. So who are we? We are a 501c3 medical foundation. We are focused on neurofibromatosis, which is a genetic disorder. There are 3 forms of neurofibromatosis, NF1, NF2, and schwannomatosis. They typically have a plethora of problems, but let's say in common between the 3 is that they have tumors that grow on the nerves, which can be extremely painful, can lead to deafness, blindness, even to cancer. Very often these people are in horrible pain, and one of the other effects, especially for NF1, is also learning issues. So the cognitive presentation was very close to my heart. The CTF philosophy is how can we change to fix the roadblocks, and we really want to use innovation and creativity to fix those problems. So quickly for the patients, what have we done? We have launched the NF Clinic Network, which gives people access to better care. We have the NF Forum, where patients are really bringing together so that we can educate and they can educate each other. There we have a lot of educational materials. For the lack of knowledge around patient needs, we have the Volunteer Leadership Council, and we're working very closely. I was very happy to see this today that there are also people from the other NF organization, which is Advocure. They are here, so welcome and thank you for being here. So we have a lot of interactions also with other NF organizations, but we have our own Volunteer Leadership Council. The limited access to clinical trials. We have the NF registry, which is a patient-entered NF registry, where we really ask questions to patients about their disorder and where we, in fact, try to connect them to potential clinical trials that are available. For the researchers, the tissue is scarce, so we launched a CTF Open Biobank, which was a interesting exercise. I was yelled at a little bit by doing that, but I sometimes am a little deaf. The small, ill-defined patient population, we're working very closely with another, which is a very innovative exercise from our clinicians, worldwide effort, where the clinicians come together and decide on endpoints. And basically, we pay for wine, beer, and food so they can keep going and come up with well-validated endpoints for our patient population. The unknown market, we have funded a market study because we wanted to make sure that when we talk to companies, we can actually tell them that, yes, you can make money with NF, so it's not a problem to enter into the NF space. The last thing is the lack of tools and cells, animal models, and also data. And then in general, the lack of researchers in collaboration, where we've worked very closely with Sage, and that is in the last example. So the lack of tools. One of the things that we have seen is that since we are a nonprofit foundation, very often people call us and say, I would like to donate my body to NF, which very often these people cannot donate their body to a hospital because if they were not treated in that hospital, they bump into all kind of IRB issues. We came up with a creative model where we collaborate with the biobank company, we collaborate with the National Disease Research Interchange, we work with these people, and then at the end also, once we get through the whole process, you will see we have also close collaboration with Sage. So the patients get consented by us, they donate their blood, the biobank is completely ready, they prepare the kits, the National Disease Research Interchange gets informed that there is a new patient, and we keep in touch with the patient. So the moment the patient passes away, the friend or family member contacts NDRI. NDRI does the tissue recovery. they sent that to the biobank in parallel. No, they send it to the NF pathologist to make sure that it is indeed NF tissue. And immediately in parallel, the tissue gets also sent to a lab in the University of Florida, who is making cell lines for the NF community. The cell lines, they are immediately available to everybody. So basically, as we go, we make the tools available. And the other tissue, once approved by the pathologist that this is NF tissue, goes to the biobank. And then once we have enough tumors per tumor type, they are sent to a tissue analysis center in Alabama. And then the data goes to SAGE. So at the end of the day, basically what we have data as we go, and we generate cell lines. The consortia science is one of my other obsessions, is how can we get scientists to really work together independent of what their expertise is. And the whole idea is how can we accelerate the path from basic science to clinical benefits? How can we understand each other's failures, and how can we make sure that those failures reinform the next generation biology? The other thing is how can we make the data public as quickly as possible, and how can we break the walls through all those, what I always think, pretty artificially divided categories of scientists, where people always ask me, what are you? And I said, I'm a scientist. And then they say, but that's not good enough. What kind of scientist? So I always make fun of them, that I think that is kind of a way to, it's a reductionist scientist of boxing up people into a block. And then the whole thing is also, Stephen and I have worked very closely together to make sure that all the data gets managed centrally. And what is really nice there also is, let's say, an optimistic thing that I'm observing is that when I started the centralized data management analysis at SAGE, people said, that's never going to work. And I was like, OK, that's fine. Now I get the opposite. Now I get calls from people that said, is there an opportunity that CTF could co-fund my grant so the data is at SAGE? Which I think is pretty interesting as a success metric. So the first consortium that I just quickly wanted to talk about is the Preclinical Consortium. That's a consortium that was launched in 2008, which are academic centers that have animal models for NF1 specifically. And they test those they test repurposed drugs, which we will hear about also in my team talk, in the people that are on my team today. So we repurpose drugs from other disease areas, specifically from oncology. We test them in the animal models, and then we can immediately go to the clinic. The interesting thing of that consortium is that unpublished data gets discussed, targets get evaluated amongst the researchers, and the clinicians are involved in that effort. So we immediately know, they can immediately be informed if something is coming their way and that is of interest or not. And we have made now an effort where we have tested a MEK inhibitor, went from proof of concept, so the first time in an animal, let's check if it works or not. Today we are in registration trial in less than 3 years. So I think this works. The second collaborative model that we have developed, that I call a collaborative accelerator, is Synodos. The idea there is to create some kind of dream team of very diverse experts, to have the data shared among each other right away and with the world after 12 months. So also the negative data gets shared with the world, because that was one of my frustrations, that I kept getting grants screening the same library over and over again, knowing that I already did it, but I couldn't talk about it. So I was trying to figure out a way to also get the negative data out there. And then the other thing is that the projects are really milestone driven. We manage that project at CTF. So people, you don't get the results, you don't get your milestone, you unfortunately won't get paid. So that helps a lot in driving research quickly. So as you can see from the slides, the fact that these dream teams are together, that's really to delay misinterpretation very often of scientists' data sets, to learn from each other. Very interesting discussions are happening at the Synodos meetings. And then the fact that the data is shared immediately and will be shared with the world 12 months later gives the researchers a sense of urgency that if you want to publish it, you better publish it now, because if you don't publish it now, the world will see it. So that's a very fast model to make sure that we get the data out there. negative data, so we stop funding the same studies over and over again. And then the milestone-driven projects, the reason why that is really important is not to make people suffer, but to really say, if you know that May 2, your cell data is going to be out, we know that and they know that, so we can program the animal study to start on May 3, which saves an enormous amount of time. So just to go a little bit more into the specifics, we have now, since early 2014, we launched Synodos for NF2. There are 12 academic centers. And as you can see from the achievements, in less than 12 months, I think we have achieved a couple of things here. That we have now a screening platform that we never had before. The in vitro screens are all finalized, and we're ready to go into the in vivo testing. The scientists are looking at potential combination therapies, which are very interesting because they're fully informed by the biology, and all the data is centralized at Sage. So this would, of course, not be possible without my teammates. So I really wanted to thank the Sage Bionetworks team, which is a pretty big team, the Synodos team, and the Synodos teams, because now we're launching 3 others for NF1, the NF Preclinical Consortium team, NTAP, who is a collaborator and a co-funder of the NF Preclinical Consortium, and of course my own teammates. So that is kind of the story of the Children's Tumor Foundation. We are doing more than that, but I thought to limit it to these 2 success stories. So that's for what I wanted to tell you. What I wanted to announce also is that in our team this afternoon, we have 3 other talks from Laura Isabel McCall from UCSD, Anjan Debnat, UCSD, and then Laurie Rumker, who is a student at Stanford. And I just wanted to say that we've had a short chat this morning. And it's interesting because Laura and Anjan, they will really present some drug repurposing opportunities in rare disease. Laura Isabel in Leishmania and Anjan in amoebiasis. I have a hard time to say that. So that is really what are repurposing opportunities? What can we do R&D-wise now? And then Laurie has taken a different approach, which is very contrasting to the other 2, which I think is very interesting, is starting with the biology. Let's start with the biology first. Let's completely understand how the systems are working in their ecology, in their ecological system, before we start to do R&D. So kind of the 2 was a very nice to get these 3 people, R&D now versus biology first and R&D later. So with that, I wanted to end here.